in our pangenomic approach, we didn't do any filters. So here we are using all the type of variation, not only SNPs. And also, if you see the association of chromosome 1 that is lower, it's because we are not working by loco, because usually GEMMA works also consider the genotype phenotype association using single chromosome. And this is what they done there on the first line. Instead, we are using the whole genome. This is the first thing. And the second thing is that, maybe I hit the slide. So the second thing is that we are finding on chromosome 3 and the chromosome Y, you can see that there's a stronger peaks. Oh. So the other thing is that repeats affect our results, because you can see here on chromosome 3 and on chromosome Y, there's strange peaks, and they are due by repeats. So if you can see here, this is an example. On chromosome 3, what you see here is the pangenome graph of this location, and this is full of repeats. So what we want to do is do some quality filters, remove some missing and some missing data, but in our case will be some missing nodes that will be not present in all samples, and mainly mask the repeats and see which will be the result. Thank you.
This looks very nice when you have a cross between two inbred strains. What if you're utilizing heterogeneous stocks and the pangenome graph has many more dimensions? Is it the first that will scale up?
I think yes, we can do this because, for example,
theoretically, or actually you can do it computationally as well, because the complexity doesn't just scale linearly. It gets.
I know. I mean, in the pangenomic world, they already done similar approach using the tool is BGGF, but I tried this on rat, and it was not working due to the high computational problem. Instead, with this approach, we will not have problem. So I done this in 2 days. So I mean, I can do that, and I'm doing this on thousands of humans, so it's working. So we'll see.
Yes, we can align, we can interact. All right, thank you, Flavia. The speaker of today, Genome from the University of Michigan, Department of Human Genetics. He's going to talk about how to study phylogeny and genome diversity in a haplotype manner way.
Sharing. It's fine. I'm just trying to
Oh, can see. Yeah, I think I'll start. So, hello everyone. I am Yan Chao from the Genome lab at the University of Michigan, and also I go by Han. So today I'm going to talk about our study in ancestral haplotypes in the laboratory rat. So the core question we are trying to address in this study is that how many distinct ancestral haplotypes are there within the laboratory rat? And to help you understand by what I mean, let's look at this genotype plot. So here on the y-axis, we have about 33 samples from 33 distinct strains. And then on the x-axis, we have about 1,000 SNPs on chromosome 19. And here, the color represents the actual genotype, with the white being the reference and the red being the alternative. So the first thing I want to highlight is that despite having data from 33 strains, we don't see 33 individual patterns. Instead, for given regions, usually only see a limited amount of distinct ancestral haplotypes. For example, here on this block on the right, we only see three types of distinct patterns. And these are what we refer to as distinct ancestral haplotypes. And for this block on the left, at first glance, you may see there is like 3 patterns. actually, there is a top row that's white. Somehow it's not visible on the projection. But actually, there's only 3 because the 2 regions highlighted by blue here are actually of the same patterns. So here, both blocks have 3 distinct patterns, but the detailed composition of these patterns changes. And the boundaries where this happen, we think are the location of historical recombination event. And one of the key things we need to do in this project is to accurately infer these boundaries. So the first thing we need to do is that we need to take the whole rat genome and break it down into individual, a few like haplotype blocks. And the boundary between these blocks are where the composition changes. And here in this example, where this happens is highlighted in this blue box, and you can see a zoom-in view on the right here. So what we need to do is we need to find these boundaries and draw the line and do this at whole genome level. So historically, we've been relying on a piece of software that's LD-based, called HaploView, which has good use, but it also has many limitations. The LD means that's for linkage disequilibrium. And recently, we have switched to some ARG-based software. ARG stands for Ancestral Recombination Graph. The specific one, TSINFER, we are using, actually, we have more success with it. Basically, we input our genotype data, which is called from the whole genome sequencing data, and along with some other parameters, such as which allele is the derived, which one is the ancestral. And then from the output of TSINFER, we can write code to plot the information we need, which is the boundaries between these ancestral haplotype blocks. So here, for example, for the whole chromosome 19, the TSINFER actually break it down into about 6,000 blocks. Now that we have these blocks defined, what we need to do next is that within each block, we need to define how many distinct ancestral haplotypes there are within each block. And then we need to assign them to individual samples. So the way we do this is, and there's the top row that's completely white, which is the reference, the 2 reference strings. So what we do, like looking at this, my brain is telling me there's 3 patterns. One of them is completely white. And but how do we implement this as computer code? The way we do this is why we convert this genotype data from 33 examples into a genotype into an IBS proximity matrix, 33 times 33. And then we can pick a cutoff line such as 95%, and we will be able to binarize this matrix. This binarized matrix, then we can assign the ancestral haplotypes for each samples. The same can be done for the block on the left and for the rest of genome. So now we have this 6,000 block, and if we do this type of assignment method on all of them, we will end up with a plot like this. So this is what we refer to as the ancestral haplotype block. And here on the y-axis, again, we have 33 individual strings. And on the x-axis here, this time it's the whole chromosome 19 or the SNPs on chromosome 19. And the color here is not genotype, but instead the ancestral haplotypes distinct. And immediately we can notice that despite we having so many strings, actually for each given regions, we usually only see from 2 to 6 distinct ancestral haplotypes. So we think this view is pretty nice, but it still has many problems. And one of the major ones is that the number of blocks is much higher than what we expected. So for example, for a small chromosome such as chromosome 19, we don't expect to say 6,000 blocks. And by looking deeper into the data, we realize what's happening is that what we are interested in is the recombination-driven blocks. However, sometimes there is mutation in the individual lines, and they also will cause the HaploView, the TSINFER, to define additional blocks. So that's why we introduced the next steps, which is the horizontal merging of the over-segmented blocks. So essentially what we do is that we treat each of the 2 adjacent blocks as one of the potential merging candidates, and we evaluate them for merging. So for each of these candidates, we calculate what we call the cost of merging. It's calculating using this formula below. So the total cost of merging is equal to D1 times L1 plus D2 times L2. The D is the discordance between the M merge block and the hypostatically merge block, and L is the length of the block. It's all right if it's not completely clear now. I promise it will be much clearer if I go through some examples. Yeah, so here again, we have 2 blocks defined by the TSINFER, the block 1 and the block 2, and you are looking at the genotype data of them. So they are adjacent to each other, and this is what we refer to like one unit of merging candidate. And what I'm going to do next is going to evaluate for if we want to merge it or not. So already know how to go from the genotype to the ancestral haplotypes. So there's 3 types here. Yeah, you really cannot see the first, which is a little bit yellowish. So, it's visible on my screen, but somehow in the projection, it's clumpy and invisible. So, so we have this ancestral haplotypes, and then what we do is we hypostatically merge these 2, like we treat this as one block, and then define the ancestral haplotypes again. And you will notice for this instance, the merge block is actually exactly identical to the unmerged block. And this is why when we compare the 2, discordance between the 2, 5 minutes grid, is 0. So D1 equal to D2 equals 0, and L is just the length of the unmerged block. The total, you can calculate the total cost of merging, which is 0 times something plus 0 times thing, and that's always 0. So usually what we do is we pick some cutoff line. So in this case, we pick 1,000. Since 0 is less than 1,000, in this case, we say we approve the merge. And then we will look at another example. So this example is block 3 and block 4, and we know how to go from the genotype data to the ancestral haplotype data. And we can already see in this case, the 2 blocks before merging, they are different. But then what we do is we hypostatically merge these 2 blocks. So for the block 3 plus 4, we'll notice that we will then define ancestral haplotype for this block as a whole. And we will notice that upon doing this, we actually introduce a new ancestral haplotype is defined, the number 4, the color purple here. So, and these ancestral haplotype number 4 is assigned to 4 samples. So now when we compare to the hypostatically merged to the unmerged one, we'll have a discordance of 4 samples, and then we have the length. Then if we calculate the total cost of merging, in this case, it will be 4 times 500 plus about 4 times 900. And the total cost of merging in this case is more than 5,000. Since it's higher than our cutoff line, so we will say that we do not approve merge. So if you notice, this total cost of merging has some interesting property. In simple case like this, the total cost of merging is actually the area of the purple triangle, or the region, the area of the difference between the merged and unmerged one. This is why I refer to it as the total cost of merging. And this is one of the merging methods we developed. This particular one is called type G. So if I apply this method with the parameter of 1,000 for cutoff to the 6,000 blocks we had before, this is what we had before applying the method. And this is what we have for after this merging method being applied. And you can see the total number of blocks has been reduced from 6,000 to 200. And we draw our conclusion based on this merged version. So finally, there's one more thing which we do. So up until this point, we assign ancestral haplotypes purely based on frequency. So the most common ancestral haplotype is assigned a lower number. In this case, the most common one is blue, number 1, and the second most common one is the red, number 2. What we find is that sometimes by lessening this rule, we can achieve higher continuity horizontally, which means last jumps and the last flickers. Why we care about that is that it is more likely for one ancestor to have a recombination than like 2 different independent ones to have the same recombination. So the individual map we can create it with the fewest number of transition or jumps or flicker will probably be the closest to the actual horizontal genealogy lineage of these samples. So without going too much detail, this is what we have before and after this method, one of the algorithm we've been applied. And we can see the total number of jumps reduced by 4 absolute percentage and the flicker reduced by 2%. So to summary, I think first our data highlights the extensive sharing of ancestral genome materials when these laboratory rat strains were created. So we analyzed the panel of 163 rats, but we find that most regions actually only have 2 to 6 distinct haplotypes. And that the 5 most common ancestral haplotypes actually make up for more than 95% of all the genome material in these samples. So we think what's happening is that even though we have so many strains, but many of them own their ancestor to the same, many of the similar panels. So for example, the Western rat outbreak, the 2 outbreak panel, the Western rat and the Strong Dolly, they probably contribute to many of the strains we are using today. And second, our analysis provide insight into the evolution of rat strains and also the information about region-specific diversity. And we think this data can be used to support haplotype-based genetic mapping and association studies. So for example, most GWAS were done using SNPs, and there's more recently, I know just talking to some people who are trying to do GWAS with more complex genome variation. It's possible that some of these complex variants that cannot be tagged with a single SNP can be tagged with a combination of steps, which is essentially what we are doing now. The ancestral haplotypes are. Finally, there's an existing field trying to array of publications. They are about sophisticated algorithms trying to determine local ancestry in the mixed populations, many of them using complex HMM model. However, there's a fundamental difference between our message and what they have. 30 seconds. Yeah, and is that we don't require a reference panel. By that, we think our message should have wider adaptability. And we are actually actively exploring what are some of the things we can do with our data and methodology. If you have any thoughts, please talk to us. We would love that. Thank you very much. And finally, I want to thank my lab, my dissertation committee, especially my mentor, Junli. And also the data we're using come from the International Ratomic Consortium. I want to thank you so much for that. And special thanks to Dr. Hao Chen for arranging the meeting and leading some of the effort. And also, I think Dr. Chen was the one who first brought the ARG-based software to our attention. Yeah, thanks so much. I will take any questions and the feedbacks.
Thank you. Any questions from the audience?
Great talk. Quick question, but if you can, because otherwise. How do you determine the cutoff for your cost conversion? How do you determine the cutoff for your cost conversion?
Yeah, the more robust approach will be try different cost conversion and then have some matrix like more than you have add or not. In this case, I did not do that. Then I have all the way from 5,000 steps all the way from 1,000 to 10,000. I still haven't determined which one is the best yet. I haven't do the full analysis I would hope for. So at this point, I just use the most conservative one we have. Yeah.
Any questions?
It seems like when you're trying to reduce the number of haplotypes, one explanation is that there's a new mutation on one of the haplotypes, right? So there's a kind of single step. But there could also be genotyping errors, right? And of course, you definitely want to get rid of all the haplotypes that are only due to a genotyping error. Do you have a sense for the ratio of those two issues in the data that you're working with?
Yeah, so actually, this is why when we use the IBS matrix, we use the cutoff. For example, the cutoff right now we're using is 95%. So if for the given region, if two haplotypes is different by 2 SNPs, but the whole region is like 100 nucleotide long, we will treat them as the same ancestral haplotypes. And also, when you look at the genotype plot, you see even though the whole pattern is very similar, but then you will always see this bunch of group with like individual pixel difference all everywhere. These are the mutation-driven things that we are last interested in.
Okay, I think it's time to the next speaker. The next speaker is Dr. Peter Doss. He came from the University of the Texas Health Science Center. He's going to talking about the genomics and the genetics of the immunoglobulins in rats.
Yeah. Thank you very much. Let me begin by thanking Anne and Mindy and Aaron and Jennifer and Mary, and I'm sure many others behind the scenes here. This is on. So,
yeah,
okay. Behind the scenes who have made this meeting possible. It's a tremendous amount of work, and I'm very grateful to them for all that they've done in bringing us together here so pleasantly. Okay. It's not a keyboard. I'm afraid your mouse has got me beat.
This one.
Oh, I use that keyboard. No, I just want to forward.
Oh, you want to use it?
Just to go forward. I can forward from here.
You can use it from,
Oh, yeah.
you can use that.
No, oh, that is just a big number.
That's right. So, some of what I have to say will contrast a little bit with the previous speaker, because much of what I'm interested in is variation that doesn't arise as a result of single nucleotide polymorphism. And the genetics and genomics of the immunoglobulin locus in the rat, as well as in other species, provides an excellent illustration for that. So let me first of all provide the rationale for the studies that I'm going to report. My lab has been interested in the spontaneously hypertensive rat. It's a model of high blood pressure in which two parental individuals were founders of the generation of this strain, and after 8 generations of selective breeding on the trait of elevated blood pressure, all of the members of the progeny had substantial hypertension. At that point, the genealogy was split into various clades, and later on the left-hand side, you see the strain that developed the bad things that high blood pressure does to you, which is stroke and kidney disease. That's the SHRSP, and we in particular used the A3 strain in our studies. The other side of the genealogy are strains that have very similar elevated levels of blood pressure, but don't get any of the adverse end organ diseases that occur in the SP lines. And so it's a great opportunity to reflect on the fact that high blood pressure is bad for you, but the bad things that it does to you depend on other genetic variation, and therefore this presents a great opportunity to uncover that other variation. And you can see on the right-hand side, we've used about 10,000 SNPs to look at how these two strains, A3 and B2, that we're studying, actually diverge, and they diverge in haplotype blocks that represent about a total of 150 blocks. But overall, the two strains are 87% identical by descent. So as a result of some studies that were going on for a few years in our lab, we focused on this block at the terminal end of chromosome 6, which is the block that contains the immunoglobulin heavy chain locus. And we used a congenic approach. The red color is the injury-prone strain, the green color is the B2 injury-resistant strain, and the blue is the congenic strain. On the top left-hand bar graph, you see measures of renal injury measured by histology, glomerular injury, tubular interstitial injury, and albuminuria. And you can see that the congenic produced by backcrossing in the B2 locus into the A3 background has improved scores for injury compared to the A3. And likewise, below it, the sort of clinical urinary markers of injury diverge and are improved. Of course, there are a few other genes in this locus, in this block, and we addressed the question of whether we could prove this was specifically due to antibodies by using a model that Aaron originally developed, which is a zinc finger nuclease targeted immunoglobulin heavy chain in the rat, backcrossed that into our strain. And you can see a similar, indeed sometimes more beneficial effect of eliminating immunoglobulin heavy chain, which means there's no B cells and no antibodies in these animals. And it doesn't work simply by changing blood pressure, as you can see on the bottom right-hand side. So we have been assembling genomes using long-read and other associated technologies. Here are genomes to make a pan-genome graft of chromosome 6. And you can see that the right-hand side and the left-hand side both show a lot of structural variation between different lines. The rest of the chromosome doesn't. And so if we zoom in on chromosome 6's distal end, we can see what the pangenome graph looks like there. So echoing what we just heard, you can see there's evidence of ancestral haplotypes in the arrangement of the genome at this end of this chromosome, and that you might expect substantial variation in immunoglobulins arising from that. So we're going to embark now on a study of the heredity of antibody inheritance. And to do so, we need to know a little bit about the structure of antibodies. You're familiar with this sort of Y-shaped structure that I have here. We're focused on the long arms here, which are the heavy chains. They're homodimers. And the constant region down below is used for downstream immune response effector activation, whereas the antigen specificity comes from the upper part of the region, which is the variable region. And so we can make IGs of a variety of different isotypes, or sometimes called classes, IgM, IgGs of different types, and IgA. And those come from a variety of different coding exons that are present in this locus. So you can see those different classes represented by colored blocks here. And I've used a tool called narwhals here, which is a way of plotting segmental duplication events in the genome. And I don't know if my pointer will show it sufficiently. I'm just going to use the faster pointer. But you can see that we have a distinct XY diagonal here. And that diagonal breaks here, and we have two parallel lines, and then it's further broken up here. So that breakage is actually taking place across these blocks where the constant regions are encoded. And you can see there's just one copy of IgM, but in SHRA2, this region's been duplicated. And it results in not only the presence of the expected copies of IgG2C and IgGD, but duplicated copies as well. So there's structural variation affecting this region. And Oops, and I now need to go back and talk to you about the other structural complexity, because we have to make antibodies that can recognize essentially almost any antigen. And we do that by having in our store a variety of these segmental gene components that are used to make up immunoglobulins. And they're used to make up immunoglobulins by somatic recombination events. So a joining segment will join with a D segment by recombination, and eventually one of the many variable D segments will come in and join with that, and then you'll get expression along with a constant region here. But it gets further complicated because not only do we have to have class switch recombination, again, that's a symmetric recombination event that occurs here, but we also get in this CDR3 region around the variable exons. That's the region where you get your antigen specificity, and that results from somatic hypermutation in this region. So one of the tools that we've used is this software that was developed by Sapanova and others to predict the presence of V, D, and J genes in the immunoglobulin loci. And we've applied that to the various assemblies that we've made, and we can see vast differences in the number of V genes that are in these high-quality, Hi-Fi-based and T2T assemblies. So we range from about 170 up to 400. And even the D and the J genes have variations in numbers. So there's segmental duplication events that have happened here. And if you contrast A3's V region, for example, it's about 4 million bases. But in B2, this segment's about 8 million bases. And it's the result of this extensive segmental duplication, which is maximized right here. So now we want to study heritability in the locus that's crazy complex and variable, and it exists to respond to antigenic input. So we know that the haplotype IgH determines susceptibility to resistance or disease, and one haplotype produces pathogenic antibodies, but the other doesn't. But we have this problem of G versus E because these are supposed to respond to environmental inputs in the form of antigens. And we also know that there are transacting effects and affect immunoglobulin production. So our approach to beginning to understand heritability in this region has to make this congenic that's isogenic for the A3 genetic background, but the IgH locus contains both, there we are, contains both haplotypes. So individuals of this type will be able to express from both haplotypes, but will always have the same environmental input within the same individual animal. So our approach to investigating this is to use isoseq, because we can't look at expression in this locus with using short-read RNA sequencing. The transcripts are about at 1.5 kb, and we can capture all of the variation in individual transcripts. And we're using the high-throughput Revio flow cells to do this. We get about 800 mega bases of IgH reads, so about 500,000 IgH reads per flow cell. And because of sequence variation between the two haplotypes, we can sort these by haplotype of origin. We can also sort them by the isotype that's present. So we can separate essentially all of the immunoglobulin reads into the different classes. So if we just look here at expressivity of the constant loci, you can see those constant loci on the axis at the bottom of the plot here, wrong button. You can see there's a huge bias for most of these, towards expression from the A3 haplotype, which is rather surprising to us. You wouldn't be able to predict that from sequence alone. Look at the V genes. We had to take these sequences and put them into this NCBI tool called IgBlast. And that gives us a readout from our different sequences where we can estimate the frequency of V gene usage. And so what I've done is pool all the IgH classes together from all of our samples here. And you can see that the blue, the B2 haplotype, shows a lot of expression from these V genes, and very little from the A3. And so there's some A3 V genes that are highly expressed compared to B2. And that's consistent across multiple samples. It's consistent for different IG classes, and it's persistent also against across time points. This is 40 weeks of age. We then have to face the, so we see that there are clear differences in expression of the constant genes, clear difference in the expression of some of the V genes, but ultimately we've got to deal with the fact that we make a specific antibody by somatic hypermutation in the CDR3 region. So that's the part that's going to recognize the antigen, and it appears to me that's what's happening differently between our two strains. When all of those processes have taken place, is there any heritability that's affecting what the outcome is in terms of antibodies that we produce? And it's a complicated problem. The CDR region is about 14 amino acids long, and we're making repertoires here. So we're mutating, we're producing different amino acids at different loci within here to give us our specificity. different clones will be amplified because they're successful in reacting to antigens and will represent a larger part of the repertoire. We're really looking at a population of sequences here. And the question is, if we want to pursue these studies any further, is there evidence of heritability in that data set as well? So we've used this immunoref analytical tool to look at the populations here by a variety of different factors. This is CDR3 amino acid frequency. So here's the 18-week A3 data, the 40-week A3 data in red. The greens are the B2 at the same ages. And what we're looking for is similarity, that is the lack of difference within these. And you can see that looking just at these CDR3 regions, as the animals go from 18 to 14 weeks of age, the CDR3 repertoires have greater similarity in their amino acid frequency. And that's true for both strains, but the amino acid frequency is different between both strains at both times as well. You can use the same kind of analysis for the Kamer frequency and see that here. And similarly with VDJ gene uses, there are also strain-determined differences. In fact, it may be that VDJ usage that's actually driving these changes here. So there are heritable differences. I'll conclude by saying we got this model. We're using IsoSeq to evaluate it, because you have to do that to have these long and highly diverse reads. And we can get complete transcripts for multiple classes. We can measure quantitative effects with this approach, because with Kinnex, you're getting enough reads to be able to do that. We can see quantitative effects of haplotype of origin in both the constant region and the variable region, and we see that that carries through to some extent in CDR3 analysis. So we believe at this point we have the sequence of those pathogenic antibodies. We just don't quite know yet, among all of the sequences we have, which ones the pathogenic ones are. But we're working on that. So thank you very much. And the people that have contributed most to this work are Melissa Smith in Louisville, who does the IsoSeq sequencing for us, Ted Kopfleisch in Kentucky, who's helped us with the genome assembly, Jiaming Zhu in my lab, who does the animal stuff. And I mentioned Jana's work in developing the IG detective tool. Happy to try to answer any questions.
Do you have any explanation for the hydrogen the two heavy chain has lower expression level than the
So we see lots of differences between the strains. In some B2 expressions are high and some are low. Probably the ones that are highest in B2 are also the ones that would have probably increased expression due to duplication events. There's more than one copy of that particular V gene in the genome. But when it comes to genes that are expressed from both haplotypes and where there isn't duplication, we don't really have a good answer for why there's this persistent difference in expression. I don't think we can resolve that at the sequence level. We might be able to resolve some of them at the duplication level, but not at the sequence level.
IgGs, which I think is black from plasma or blood, could feed the two animals to see whether or not that was sufficient to either give.
Yeah, so we have, in fact, the problem's getting, and so it's best actually done in the knockout where there is no immunoglobulin present, where you can actually do an even more controlled expression, which is to give the knockout either serum from the injury-resistant strain or the other strain. We've been doing that for a while, but we need to keep doing it because the effects are relatively small, and we're measuring relatively imprecise measures of renal injury, histological injuries measured by a human's eyes. But we're getting there. I think that we may be able to show that in
Oh. Yeah, I came up with
Okay, yeah, I think we need to move to the next speaker. So next speaker is based on Okamoto.
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 Oh,
That's correct.
Okamoto.
Okamoto.
Oh, yeah, it's
Oh.
not
Okamoto.
It's an O sound.
Okay. That's from the University of California, Experiment. Yeah.
Not Davis.
Oh, Santiago, sorry.
Davis rejected me, okay?
your turn.
Hi, I'm Faith. I'm here with an affiliation from Dr. Palmer's lab in UC San Diego, although I'm actually not working there anymore. I'm a PhD student at Santa Cruz now. This is my undergrad research. It was published in September, actually, really recently. So this talk is essentially an advertisement to get you to read my paper. How do I make this go forward? Oh, here? There we go. Anyway, okay. Okay, so this is a general roadmap for the talk. I'm going to cover my main scientific question that was driving this work, some of the data that we used to answer that question, and then the first slide said this was Y mitochondrial chromosomes. So the first step was to figure out what kind of chromosomes I have, which was calling haplotypes. Some other people here have been doing haplotypes. And then I use those haplotypes to do fun things. And then we have a conclusion slide because you have to do that. Anyway, so the main question here is, do the Y mitochondrial chromosomes, the genetics in them, affect the traits that we study in HS rats? So HS rats, that stands for heterogeneous stock. I'm the first HS rat person, but there's going to be a bunch of others hiding among the entire program. Heterogeneous stock rats were created by interbreeding 8 inbred strains, and then they've been crossed for over 100 generations now. So they are lots of recombination events everywhere, except Y mitochondrial chromosomes don't have recombination. I'm dealing with the easy chromosomes because I was an undergrad. So the mitochondrial chromosome, as you may have learned in biology, I actually forget what this is taught, is inherited only from your mother. All the sperm mitochondria are killed, and then the Y chromosome is inherited only from your father. I looked at both the Y and mitochondrial chromosomes, but this whole talk is only going to be the mitochondria because we don't have enough time. Please read the paper if you want to look at the Y chromosome story. And the reason that we're looking at this is we have been studying these rats for 10 years. So there are lots of phenotypes that we have. Mostly we've been doing GWAS of them. But the GWAS has been throwing out all of the Y mitochondrial genotypes because you can't do GWAS when the fundamental idea there is you're looking at recombination. Which means that now we want to see if those data we are throwing away are useful or not. Was this something we were missing? What I have is we have pretty good genotypes for the founders, 40x coverage. We have pretty sucky genotypes for the HS rats. They're 0.25 coverage. But it's good enough. Hey, it's good enough. And then we have a bunch of random phenotypes, which would usually collected from different rats, but there's like 4,000 or 5,000 of these things. Most of them are metabolites. But we also have a bunch of behavioral traits like how addicted they are to cocaine and whatnot. And so step 1 of the paper is find out, we have 8 founder strains. How many mitochondrial chromosomes were there in those 8 founder strains? What diversity did we have to start with? And the answer to that is not very much. There were 3 major haplotypes. ACI technically is 1 mutation off the other haplotype. Who cares? It's basically the same. There's BN, which is the reference, WKY, which is not the reference, and then there's this giant group of 6 that I'm not going to say because it would take too long. And so the answer to this question is there are 3 haplotypes, and they're basically all the same distance apart from each other, actually. And now, which of those have persisted into the modern rats? Because, again, 100 generations of breeding, we have genetic drift. And the answer is we lost one of them. I'm sorry, I didn't do that. It wasn't my fault. You can see that this is a graph of the genotypes of these SNPs. I threw away all the ones that were MAF0, so these are only the SNPs that exist in modern rats. And the higher it is on the y-axis, the more reference SNPs, and the farther the x-axis, the more alternate SNPs. And you can see there are basically 2 groups. People are either reference or they're alternate. The reason that they're not completely matching is because, again, 0.25 coverage, we are missing some SNPs. I'm sorry. That was not my fault. And you can also see the WKY was lost. It had some reference, some alternate of these SNPs, but no one looks like it. So we're dealing with mitochondria 1, MT1, and mitochondria 2, MT2. Okay, do MT1 and MT2 affect anything that we care about? So the first step was use all those phenotypes that we were using in GWAS. So these are, again, metabolites, and microbiome stuff, but also a lot of behavioral physiological things. For example, some people think that mitochondria affects, I don't know, rat strength. Do we test? We do have like length of their bones and whatnot. Anyway, so we separate the rats into two groups. This group over here has all the mitochondria 1, this group over here has the mitochondria 2, and you say, do these two groups have different phenotypes? And the answer is no, because you can take all the p-values they generate from this, and you say, what did I expect this to look like under the null hypothesis? And it looks like that. This is a QQ plot. We plotted the actual versus expected p-values, and you can see that they basically are the same, which is a great thing, because it means that all those Y mitochondrial genotypes that we were throwing away for the previous 10 years of GWAS actually didn't matter. So this is the yay result, even though it is really, really boring. Okay, this is the step where we go, well, maybe it was no hypothesis because I called all the haplotypes wrong. Theoretical possibility. But yay, it isn't. So I looked at, do these two groups of rats, my MT1, my MT2 groups, do they have different levels of gene expression? And the answer is yes. 4 mitochondrial genes only. Nothing else came up significant. And this is the best example. You can see that I have mitochondrial 1 in one side, mitochondrial 2 on the other side, and the y-axis is the expression of this mitochondrial gene. It's in the mitochondria. I don't really know what else to tell you about that. But you can see that one of the mitochondrial groups has much higher expression than the other. This one was actually replicated across like 7 of the 10 tissues I had, and a bunch of other mitochondrial genes were replicated were just in the brain group because we have the brain data set was the biggest, so I left the most associations from it. But the fact that the 2 mitochondrial groups have different gene expression is good because now we can be pretty confident that I actually called them correctly. So that this is an indication of real data saying that there really isn't any association between mitochondria and these phenotypes because we know that there is association between mitochondria and these phenotypes. And well, I think I've been talking way too fast because he only just gave me that 5-minute sign. This is near the end of the presentation. Well, I can give you some more. So the conclusion slide. See, this is why I'm thinking I'm talking too fast. But I can tell you a bunch of random stuff that's not in the slides because I thought this would take longer. Anyway, so the first conclusion that we have is there wasn't much mitochondrial Y diversity in the HS rats. You didn't get to see it, but for the Y, everyone started with a different haplotype, actually. All 8 strains of different haplotypes. There were 2 groups that they fell into, and the modern Y rats basically have 2 of the haplotypes. There's minor variation elsewhere, but only 2 haplotypes in the Y survived, and only 2 haplotypes in the mitochondria survived, which was actually great for me because most of the statistical tests you can find easily want you to have 2 groups. It's harder if there's multiple. And then those haplotypes can be very easily called from the sequencing data because, again, my haplotyping method was literally, are there more reference or are there more alternate SNPs? Because that just separated them so cleanly. I was so lucky with this. And we know that those haplotypes were called correctly because they show up in the gene expression data as actual separate groups. There were 2 genes on the Y chromosome, which was separate if you looked at the Y chromosome haplotypes. And then also these haplotypes that we can correctly call, thank goodness, have very little effect on the traits that we study in the HS rats. So all those fun alcohol phenotypes and whatnot, your Y chromosome doesn't affect how much you like alcohol if you're an HS rat. Yay? I don't know. People have looked at these kind of things in humans and the associations replicate really weirdly. Like they don't always work, maybe it's just the population group you're working with. And the main limitations of this is, again, I am working with a very specific population, so I can't say anything about what these haplotypes would look like on other genetic backgrounds. I can't say anything about the haplotypes that we don't have. WKY, there was actually a paper published this year or last year that showed that the WKY mitochondria does things differently from the B end mitochondria, but I didn't see that because I didn't see any WKY mitochondria. Again, not my fault, genetic drift or maybe selection, who knows? And we also only have the phenotypes that we have, although that's not a limitation because there's, again, way too many phenotypes. By the way, all of my data is public. It had to be, that was a journal policy, but that means that if you want to play with a giant table of phenotypes, all tied to RFIDs, it's there. You probably don't know what any of the phenotypes are, but you can look at the table. Yeah. There's a bunch of other fun little stories if you read the paper. Again, please read my paper. I would love for you to read my paper. For example, there's one of the Y chromosome genes has this weird group of low expression, and I dug deeper into that. I found out it's because there's a giant section of the Y chromosome deleted in a bunch of the rats, but I couldn't do anything with that because 0.25 expression. Some of the rats don't have any reads there because low expression. I'm sorry, it's not my fault. Yay, look, Tim, this is longer than the conclusion slide, so I just... Anyway, so these are my acknowledgements. That's the old center, the P30 thing. The P50 was starting now, and so that's where I got all the data from. There's a really, really long list of people who are authors on this paper because I took all the data, so I put them on the paper. And Palmer Lab is where I did this. Thank you, Dr. Palmer, so much for giving me an undergraduate research experience. It was great. You also paid me. That was great. Especially thank you to Apurva Chitre, who will be presenting today, yeah. And she was my mentor, and I brought her a lot of really simple questions about what I was doing, and she pushed me in the right direction. And that's a QR code to read my paper. Please read my paper. Thank you. That's all I have to say.
That was fascinating. I have three questions. First of all, can you bring reprints?
Oh, we got to know.
The journal is not, it's only an online journal. It was published in GB. So, I mean, you can print out the ticket if you want. It's a QR code. You can go on the website and download it.
Second question, in the genomes that we would sell, the mitochondrial genome, but my recollection is that none of it affects coding sequences of mitochondrial genes. So the difference in the expression you've seen may be interesting because non-coding sequence variation in the mitochondrial genome, which probably hasn't been studied very much at all, may still have an important role in the population. Is that correct? You didn't discuss what the variants that you were...
Some of the variants look like they had coding effects in the mitochondria, but I think, yeah.
The third question gives you an opportunity to talk about the Y chromosome. So, Y chromosome assembly has been a problem. Representation. You have in the work that you're doing, I'm sure you have the differences that you have are called the real differences. But I'm interested in the Y chromosome. People who study mice, for instance, don't recognize that some inbred strains of mice tend to have litters that are biased towards males or towards females. So hereditary traits are independent, and it's called sex chromosome competition or sex drive. And how it happens is interesting because we know that many of the genes on the Y that are expressed are transcription factors, and many of them are also duplicated. We'll probably be hearing about SRY later on in this meeting and how many copies of SRY are in the genome, how many might be expressed from that genome. But there are other like GSTY and USTY, where there are multiple copies of them. So I became a little interested in this because in our genome assembly in the rat, we found these areas of the autosome that were highly amplified, duplicated over and over again. And what they were duplicating were genes that were largely expressed in testes. Okay? So one way of having this sex divergence in the progeny would be if Y chromosomes, for example, carried multiple copies of one transcription factor that gives sperm with that Y chromosome an advantage because they're driving expression of all these autosomal genes that make them better at winning the race. But another pathway, maybe you have the same transcription factors, but their targets in the genome are just amplified. So where are all the phenotypic data that you've collected? Have you collected data on the sex ratio of progenies from your various crosses?
That data does exist, but it's in the pedigrees, and the pedigrees are not correct. Like
Yeah, it wasn't in the data that Faith had to analyze, but I have long been aware that we have both litter size data and sex ratio data, and it's all recorded. And, you know,
Well,
I did look at litter sizes at one point, and it wasn't significant, but that was an older version of the data, so I never looked at it.
and Peter, if you'd like to come and do a postdoc in the lab. San Diego.
is that snow or sand?
That's a holiday party. This is the last picture with me in it.
Okay.
Did you
Is there any
have-
other questions?
I feel like I should know, but I'm wondering if you sort of try to compare what you found in the genetic data with what is known about how the different strains evolve. Like, does it make sense that you would have WKY and BN on one side different from the others, and WKY, why was it lost? Was the founder a male, for example, things like that?
There was actually a previous paper that did what HS-RAF mitochondrial phylogeny. They used data that had a sample name mixed up, but other than that, they found the exact same relationship. So this, and I tried looking at the giant circle phylogeny chart of all the bat strains, and it looked, I don't know. I didn't find anything particularly interesting comparing that to my chart, but maybe I'm just not very good at doing phylogeny. I know the names of all the HS bat strains, and that's about it.
Yes, thank you.
We left a little bit of buffer time at the end of these, just in case people ran over. So if there's any other general discussion or questions on any of the talks, we have a few minutes. Otherwise, we can break early and have a little bit longer break. So your decision. Is anybody have any comments or questions about any of the talks this morning?
Actually, I have one more question about this talk. Yeah, so for my next slide, I was looking at the paper, I can't remember but looking at the figure for Y chromosome. So I see that you also only have two haplotypes for the Y chromosome. And actually for my own analysis, I actually find three haplotypes. And I looked at the age name. I think the examples in the HSHK is probably not the third haplotype analysis. So
There were 8 haplotypes among the 8 founders, and now there are only 2 because we lost 6 of them. And I can't tell you why.
okay.
But I'm very confident that those 2 haplotypes are different. And in the very in the supplements is this chart of how the versions the bats were from their own haplotypes, and there's very little variance from the consensus sequence. One haplotype has like a single stint, and some people so like 88 of 2,000 mass were different. And the other haplotype has that giant deletion variant that knocks out the entire gene. But other than that, there were not significant differences that bats had from their consensus sequences. So there are just 2 consensus haplotypes.
I see. I just have one more question. For the mitochondria, it seems like your figures in the tree says that there is 3. Why do you see there is only 2 haplotypes?
Because there were 3 founder haplotypes, and then we lost one of them. And so now we have 2. In 100 generations of breeding, genetic drift or inadvertent selection made one haplotype go to extinction.
I see. Thank you. Nice comment.
Thank you.
Any other questions? I don't know if we have any online. I can't make it through anything. Let's see.
Yeah, no more question
No
online.
more questions.
I guess an online question for you, so.
Yeah, if anybody has a question.
I have another question. It's a little opposite, not really answerable. Uh, As we've been assembling rat genomes, we've encountered a problem in assembly that arises because the nuclear genome is. I'm sorry. The nuclear genome is full of bits of the mitochondrial gene. They're called MUMT, nuclear mitochondrial sequences. And what I haven't been able to find out is whether there's any evidence that any of those might be expressed on the nucleus. When you find divergent expression of mitochondrial genes, are you finding divergence in mitochondrial expression of those genes? Or is the divergence coming because there's nuclear expression as well?
So that's what typically.
I can say something that I know.
Okay.
I found divergent expression of mitochondrially encoded mitochondrial genes did not show up in my differential expression.
And I think, Faith, you also counter-mapped those to make sure that the genes that were different based on mitochondria didn't map anywhere else in the genome, right? Oh, really? Well, we can do it in 2 minutes on that. Here we can have an answer for you in about 90 seconds. No, you need to look at the sex ratios. That's going to be your first post-op. Should you accept it?
Well, given the work I'm doing on image-1, and I'll need a new project soon.
There's no more questions. I don't see any online. We can break a little early. Please help yourself to more food if there is. I'm still recording.
It is now.
Yep. Thank you. You're on top of
Hi guys, we're on our obesity session. So our first speaker is our Chifa from the Bomber Lab. And she's going to talk about genome association with adiposity and HS.
So hello everyone, I am Pura Chatre and I am a PhD student in the bioinformatics program at UC San Diego, and I am in Abraham Palmer's lab. And the study that I will be talking about today involves heterogeneous top rats. So I wanted to give a quick recap again. And so in 1984 at the NIH, 8 inbred rat strains, which are shown here by their abbreviations. So they were interbred with the goal of creating an outbred population. So you can imagine that after several generations of crossing, you get a population that is completely outbred. So at any given locus, a given individual might have inherited their alleles from these different founders. And so each animal is genetically and phenotypically unique. And so this genetic diversity is a powerful tool for performing genetic mapping for complex traits. And so the HS rats that we've utilized for this study, they were bred at the Wake Forest University. And this colony is maintained by Dr. Leah Solberg-Woods. So our main goal is to associate the genotypes with the phenotypes, and this approach is also called as GWAS. And so before I delve deeper, I wanted to quickly mention the LIDA Center for GWAS in outbred rats. So the main goal of the center is to look at the genetics of drug abuse in HS rats. And so as part of the center, we've distributed HS rats to different phenotyping groups. And the Palmer lab has also performed the genotyping and GWAS for these different behavior traits. And so as part of the center, we have these different collaborators, including 2 labs in New York. And so they have measured hundreds of behavior traits which are related to drug abuse. But at the end of phenotyping, in order to fully utilize these rats, they have also measured different physiological traits. So the rats were fasted overnight, and different adiposity measures, including visceral fat pad weights, were also collected. And so in this table, I have listed the differences across these different phenotyping sites. So as you can see, some of the centers utilized adolescent rats, whereas others utilized older rats. There were also some breeders in one of the phenotyping sites. They were housed differently across some of these sites. And some of these rats also received moderate amounts of drug exposure, like nicotine and cocaine. And the diets were also different. So there is a lot of heterogeneity across these phenotyping sites. And so the combined sample size, like the pooled in sample size from these different sites, is approximately 8,800, also making this one of the largest rodent jivas ever performed. So once these rats are phenotyped, at PharmaLab, we've genotyped these at millions of markers. I wanted to quickly go over our GWAS approach. And so remember I mentioned that there are a lot of site-specific differences. And so in order to account for those, we've used the sites as covariates. And so we quantile normalize the phenotypes and then we pool them together and we perform GWAS. And so for GWAS, we've also used a genetic relatedness matrix to account for the complex relationship in this HS rat population. And for GWAS, we have calculated the genome-wide significance threshold using permutation. And for our downstream analysis, for calculating the SNP heritability and GWAS, we have used the software GCTA. Right, so now moving on to the results. So these traits exhibit high SNP heritability in this population. And so the lowest SNP heritability estimate was for fasting glucose. The highest was for body weight. So we can also look at how does one phenotype relates to another in observed values. And so that's the phenotypic correlation. And genetic correlation is the shared variance between the two traits due to genetics. So one thing about genetic correlation is that we can estimate it even if the traits are not measured in the same set of individuals. So here in the upper triangle, I am showing the phenotypic correlations. And the lower triangle depicts the genetic correlations. So the strongest estimate was for phenotypic correlation was between length with tail, length without tail, and the weakest was between perimetrial fat pad weight and fasting glucose. So here we have no estimate for phenotypic correlations because epididymis fat weight and perimetrial fat weight are sex-specific traits, so they're not measured in the same set of individuals. So the highest genetic correlation was observed between perimetrial fat weight and retroperitoneal fat pad weight, and the lowest was between length without tail and fasting glucose. Okay, so now moving on to finding if there are specific chromosomal locations which are associated with these different adiposity traits. So here I am showing a combined Manhattan plot for these 7 GWAS runs. So this plot is also called as a porcupine plot. And so with this sample size, we identified 116 independent QTLs for 7 adiposity traits. Alright, so on this slide, I am only showing the Manhattan plot for body weight. So this trait stood out in this data set because of the large number of QTLs we identified, and it's also the trait with the highest SNP heritability estimate. So now we wanted to take advantage of this large sample size, and we wanted to investigate if the body weight QTLs are sex-specific. So what we did is we performed a sex-stratified GWAS. So we essentially split the whole data set into males and females, and then we applied our standard GWAS pipeline separately for each sex, and we ran GWAS. So this is the porcupine plot, the combined Manhattan plot for the 2 separate GWAS runs. So the blue QTLs are for females, and the yellow ones are for males. And so we identified 14 QTLs for females and 10 for males. So I want to point out that many of these QTLs were unique to each of these sexes, and there were some which were shared across the two analyses. So this locus does look very dense, but this was unique to the male Chivas that we ran for body weight. And I want to point out that when we performed variant annotation for all the SNPs in this QTL interval, we found a high impact coding variant, splice donor variant within this gene, which is SLC11A2. So this SNP potentially alters the mRNA splicing and probably significantly alters the protein function. And so in humans, the same gene has been linked to obesity and iron accumulation in skeletal muscle. So we also wanted to perform FIVAs and see what other traits show association at this exact dot SNP. And so for this same SNP, there were different muscle phenotypes and also bone phenotypes which were significantly associated. And so this probably means that there's something about this locus that is influencing these different physiological traits, possibly through shared biological mechanisms. So this locus was the one that we identified for both males and females, and also when we did the combined analysis. And so within this QTL, we identified a high impact start-lost coding variant within the gene PR8HR. And so this variant alters the binding of the protein sequence, so it probably alters or disrupts the protein translation and function. So this gene, like the multiple rare variants within this gene, have been linked to severe obesity in humans, kind of supporting this gene's role in energy homeostasis, and it's also potential as a weight loss therapeutic target. Additionally, in 2018, Leah's group also performed Jivas for body weight, and that was in a different cohort of ages rats. And so they also identified this locus. So we've successfully replicated this finding in this data set. Right, so in conclusion, this is one of the largest rodent Jivas ever performed. And so we performed sex-stratified Jivas, and we identified multiple unique and common loci for males and females. And we've replicated previously identified loci, and we've also discovered new findings. And so this result kind of demonstrates the importance of large sample sizes for dissecting complex traits. And so we are also phenotyping additional rats. So we plan to add 1,000 more samples to this data set. And so once we have that full sample size, our main goal is to perform sex as an interaction term GWAS. I'd like to thank everyone from the LIDA P50 Center, mentors Abe and Aksana, and everyone from the Palmer Lab, and thank you all for listening.
People hear me? No, okay. No, there we go. That was a great talk. I am curious, how many animals do you need to do census or any type of right, in early days, we used to say 1,000 animals would be enough, 1,500 would be enough. But when we published in 2020 with 3,000, that I remember asking you guys, wasn't quite enough to find those size. So have you done any type of power analysis to specifically ask that question?
No, we haven't done any formal power analysis, but I remember for the 2020 data set, when we did split it for males and females, we did identify some loci, but it wasn't these many.
Thank you. Real discouraging. It's not easy to go to NIH and say I need 8,000. Yeah.
Very nice to see this study. I was thinking when you were showing the results about heritability, that since you've got such good sample sizes, I wonder if you could do something that's like what they do with polygenic risk scores using the actual detected loci to see how much you can predict It's that way. And the money that predicted.
Thank you. I think we discussed we planning on trying this out. Did you want to add anything?
Yeah, I mean, that's sort of what I guess.
Yes,
Right.
yeah.
There'll be like 5
Yeah.
minutes about it.
Okay.
So I work in laboratory, not training, but from previous experiments that I've done, I found that heritability or body weight increases with age. Actually, and so I'm wondering if that might be helpful to you in terms of finding associations with larger sizes and potentially for another subject.
So we could probably split the cohorts, like the ones which have older rats, and probably check that out. Thank you.
I have a quick one. Did you say what was the genetic correlation between males and females?
It's pretty high, but I don't have the estimates.
But it's like about 0.8 or something like this, or 0.5 anything.
0.5. Thank you.
Yeah, that's all we would, with her study identified. Gave identification of teams that were obesity or national.
Do you prefer this or that? Oh, yeah. Okay with that? Hold it up. Oh, yeah. Can people hear me now? Perfect.
You hold it between your teeth, that would be.
I will not do that. Yeah, right. Germs are everywhere. So I would like to thank the organizers for inviting me to speak today. It's so exciting for me to be back. For those of you who don't know, I used to work at MCW for more than 10 years and left in 2016 and haven't been back until now. So it's just wonderful to be here. So I am going to talk about work in my lab where we've used outbred HS rats, which you've heard about, to identify genes for obesity, and have subsequently found that those same genes are also playing a role in emotional behaviors. But either one will be fine. Yeah, thank you. So I think all of us know that both obesity and mental health diseases are increasing. I've put up 2 CDC maps, one in 2013, the other one in 2023, that's for obesity. And as the colors get darker, that means the prevalence of obesity is increasing. By 2030, it's expected that most states in the United States will have about 50% of people who are obese. The slide on the, there we go. The slide on the left or right is periods of first major depressive episodes over time, and it's split by age group. So the 2 in blue are for under age 26. And you can see starting in about 2012, that's starting to really skyrocket, and it's gotten worse during COVID and after COVID. So really important diseases that are getting worse over time. And what we know is that obesity and mental health disease are bidirectionally associated. And what that means is if you have one, you are more likely to have the other. And there are all sorts of things that go into that. So in terms of environment, diet and exercise play a big role. We know that inflammation plays a big role, but genetics is a really important piece. And there's actually relatively few genes that have been identified linking both diseases. So that is something that this talk is going to do. So this is the outbred HS rat. I am not going to go into detail here, but a shout out to Abe Palmer, who has just been funded for a P30 for a Center on Genetic and Epigenetic Study of outbred rats. So
They're great for genetic mapping, and that is what we have done. Okay, so for this study, the two statisticians that I have worked with, because I am not a statistician, are Will Valdar and Richard Mott. And these two peaks are probably very familiar because Apurva just showed them. Chromosome 1 and chromosome 6 are two loci that we have seen basically in every study that we have done. And the more animals we have, the higher those peaks go. So something very real is going on in those regions. I am going to talk about the region on chromosome 6. And we found 2 genes in that region, and I think there's probably more. So the 2 genes that we've identified in different ways. CRTCAP3 was identified using mediation analysis. And so what that means is that it's expression levels of CRTCAP3 that are likely driving the adiposity phenotype. And the second gene is ADCY3, where we identified a non-synonymous variant. So I'm going to start, and I'm going to talk about CRTCAP3. This is a gene that when we started, there was very, very little known about this gene. And so the first thing that we set out to do is to create a rat knockout. We partnered with Aaron Gertz, or Aaron Gertz did this work for us. And it was basically a 2-base pair deletion. We used the Worcester-Kyoto rat as our background, and that was based on the haplotype data in the HS rats. So all the work I'm going to talk about was done by a graduate student in my lab, Lexi Salanci. She's since graduated. But the initial hypothesis is that we knock out CRTCAP3 expression and retrofat pad is going to go up. And for those of you that don't know, that's a visceral fat pad right by the kidney. And that's exactly what we found in our first study. So here I'm showing females where we saw a stronger phenotype. The knockouts are in gray. So you can see their growth curves. The knockouts are heavier, fat pads are heavier in the middle, and it's because they ate more. So we were very excited about this finding. We did a second study so that we could dig more into it and figure out what was going on. Why are these rats bigger? And this is what we found. So we didn't replicate it. Lexi was very upset about this. This was meant to be her thesis. But like good scientists, we were like, okay, what could be going on? What is different? Was the first one a fluke? Maybe it was a fluke. Sometimes they're a fluke. Maybe something more was going on. And one of the things that she found is that the wild types changed between the 2 studies. So here, this first slide is study 1 versus study 2 in the wild types. And you can see that the wild types got bigger, a lot bigger in study 2. There was not the same change in the knockouts. And you can see that the ones on that side are the fat pads. So both body weight and fat pads got bigger in the wild type. So what was different? What could explain this? There was a really, really, really big environmental difference between study 1 and study 2. Study 1 was done before COVID-19 happened. Study 2 was done during the COVID-19 shutdowns. So before, we had construction right next door, vibrations in the building, we had parties in our building right next to the vivarium. That's great. and we also had lots of foot traffic, right? COVID-19 shutdowns happened just as our animals were being born. We were going to continue this study anyhow, but everything was quiet, right? The building is shut down. Only 1 or 2 people can be in at a time. So the environment that those animals had was wildly different than the first environment. So I reached out to my PhD advisor, Dr. Eva Redeye, and I said, could this be stress? We know that noise impacts stress in animals. And so we sent some samples to her because she was doing corticosterone assays, which things I did as a graduate student. And you can see here that there are pretty drastic differences between the knockout and the wild type, as well as between study 1 and study 2 in the wild type. So maybe this gene is interacting with stress to cause the adiposity phenotype. And so we did a third study. And it's a little bit complicated. It's not as clean as we wanted it to be, but I believe that this study supports the hypothesis that CRTCAP3, when it's low, interacts with stress to cause adiposity. So in this study, we started out with a noise stress, and then we did unpredictable chronic mild stress. And what you see here is that the control animals are on the left, the solid bars, the stress animals are on the right. And you can see that when you're a knockout and you're under stress, your adiposity goes up and your eating goes up. There was no change in the wild type. We also looked at a couple of behaviors. Both of these are essentially an animal, the novelty suppressed feeding, you fast the animals, and then you put a little piece of food in the middle of an arena, and a very brave animal is hungry and is going to get that food. If you're very anxious, it doesn't matter if you're hungry, you are not going to go to the center to get that food. And you can see that the knockout stress animals are not having it. They're not going to the middle. And with the open field test, again, they're not going into the middle. No differences in the wild type. So we then asked the question, well, very little is known about this gene. Where is it expressed? And all the fat pads are on the left-hand side, where it's very, very little expression. Where you start to see high expression is the pancreas, the gonads, and the gut area, so the ileum and the colon. So all of those tissues are known to be involved in the stress response. So it's possible that KIRK-CAT3 is acting in the pituitary or the colon to influence the phenotype. Okay, I need to move away from this study, but I believe that the studies are consistent with the hypothesis that low KIRK-CAT3 leads to increased adiposity under stress, and maybe it's acting in the pituitary. We have a lot more to do to figure that out. So the second gene I'm going to talk about is ADCY3. Here in the HS rat, it was the WKY haplotype that led to decreased fat mass. When we looked at the founder sequence, we found a leucine to proline in the WKY founder strain only. And so I reached out to Jeremy Prokop, who also used to be at MCW. I don't think he's here today, which is sad. But he did protein modeling that supported that this amino acid change is likely damaging. So what do? We reached out to Aaron and said, Aaron, can you help us make another rat model? And he did. So in the red box is the proline. Remember, it's the WKY that has the proline. We wanted to change it back to a leucine. It was early days of CRISPR, so they ended up changing the neighboring amino acids, which initially were like, oh, but we saw the phenotype, so we're like, okay, we'll take it. They're all in the transmembrane region, which is what we think is the important part. So all the studies I'm going to talk about here are from Mackenzie Fitzpatrick, a phenomenal graduate student in my lab. And the hypothesis is that the mutant rats are going to have increased fat pads. And that is exactly what we saw. We saw it in both the males and females. So there are the body weight curves, and it's all due to increased fat mass. There's no difference in lean mass in these animals. And we have replicated this 3 times. This is a very strong and beautiful phenotype. So the first thing that we did is ask the question, does this amino acid change cause expression level changes? And you can see here, we looked in 2 tissues, the retrofat and the hypothalamus, where this gene is very highly expressed. And there's no differences. There's no differences in RNA on the left, and there's no differences in protein on the right. So whatever it's doing is very different than just changing expression. So this time we could do a second study and try to figure out what's going on. We put the animals in the TSE chambers, where you can measure indirect calorimetry and you can measure food intake and several other things. And what you can see here is on the top is the males, and you can see that the male mutants are eating more. So that's partially explaining their increased fat pad size. There's no difference in the males in energy expenditure. If you go to the bottom, the females are not eating more, but they have decreased energy expenditure. So this is fascinating, because what this is telling us is that the same mutation that's causing the same phenotype, which is increased adiposity, is doing it differently, depending if you're a male or a female. That is cool. So ADCY3 is also known to play a role in major depressive disorder. It's been identified as a blood biomarker, and the ADCY3 knockout mice have anxiety and depressive-like behavior. So we asked the question, does this mutant cause the same thing? And so the forced swim test is a measure for despair-like behavior or passive coping, depending on which camp you're in. And for those of you that know anything about the WKY rat, they are a depressed strain. So when you have counts of immobility up at 40, that's really high. Most strains are down around 10 or 20. So I was a little bit dubious, right, that we would see any differences. The mutants are crazy immobile. And I didn't believe them. Can I have 1 more minute? Yeah. Okay. I didn't believe the people who scored this. And this is what I did as a graduate student. I scored 500 rats in the FST. So I scored them myself. And this is real. Those animals are crazy immobile. On the other side is the open field test, where you can see that the mutants have decreased rearing, which would be a type of anxiety. So now I'm on to the summaries. Mutation in the transmembrane domain of ADCY3 leads to increased adiposity in both sexes, increased eating and emotional behaviors only in the males. And so the overall summary is that the genes we've identified for adiposity are also playing a role in emotion behaviors, which is very exciting for me. So that's what I care about. And we are digging into these to figure out why. So I have lots of people to thank. Dr. Lexi Lancey did the CURT-CAT3 story. Mackenzie Fitzpatrick did the ADCY3 story. None of this work would have been possible without Aaron and Dr. Reda for all the discussions on the CORT hypothesis. So thank you. Sorry, I went long.
Whenever I think of obesity, I think of you guys, I know there's a great talk tomorrow, like, it's, I think it just touch on that. You now have use of less codes for the management stocks. Are you monitoring it? Are you maintaining it?
So in our studies, we have collected the fecal boli so that we can do that. And I know that an AIDS group has, you've partnered with people who are looking at the microbiome. We have not compared the differences between the two colonies, but we absolutely could.
It also makes me wonder about the making of the knockout as opposed to the HS, right? Because those are very different. You could transfer that, but
we could. It's worth looking at. I think it's worth looking at. We haven't done it.
You can put your mouth. Well, you're switching now.
Sure.
Thank you.
Yeah.
Okay, right. And studying college metabolism and choose with this part.
Thank you. Everyone, nice to be back at CTC. I've been here in a few years. I'm very excited to talk about metabolic traits, how they might be mediated through transcriptomes. This is work at the Jackson lab. And
You'll need to click the arrow.
yeah, okay, got it. Now I should do that. Okay, I'm going to begin and end my talk with human genetics. There won't be any in the middle of those, don't worry. Type 2 diabetes has over 250 nice GWAS associations now. Most of these are non-coding, like most of what we see in the human genome, or at least suspected to be. So there's a lot of speculation and work about how that variation translates into these ultimate disease risk phenotypes. And so we went to some mice, which are very small, not very smart rats, and use them to create complex populations and study metabolic traits. So here are the 8 founders of the collaborative cross and diversity outbred. I'm mostly going to be talking about diversity outbred here, which are outbred animals derived from those 8 founders. Animals were yanked from the CC inbred breeding pipelines, partially inbred. We had randomized breeding, and now we get these heterozygous mixed outbred populations. That's an example genome from a fairly early generation. We're on generation like 50 now, so the recombination is maximally achieved by this point, more or less. If you look across these diversity outbred mice as a genetic model of metabolic traits, you see a lot of nice heritability across a number of traits. These are mostly insulin function and adipose-related traits across the board. Pretty good heritability, nice correlation structures across those traits, as one might expect. This is primarily work that was done by Alan Addy's group and Mark Keller at Wisconsin. We leveraged this population and a lot of this in vivo data to further those studies. What the the Wisconsin groups, can't call them Minnesota, we get in trouble here. What the Wisconsin group found in collaboration with Barry Churchill at JAX was that there's really nice genetic mapping in this population. So this is across many traits. We're looking at lab scores, and seeing a variety of nice significant hits across the genome. Some of these are shared across different traits, some of those more distinct, just showing that we can map those traits. And so similar to the human population, we get plenty of genetic associations in these mice to disease-related traits. And so now the question is, can we dig in and start to understand some of the molecular processes by which we could investigate these effects? And so for that, we went to Allen NH Freezer and pulled tissues, adipose, skeletal muscle, and liver. And we did RNA-seq. They had already isolated pancreatic islets in a heroic effort in the Addy lab. And so we also did RNA-seq on those. And so we have 4 key tissues here from the same mice that they did those genetic analysis studies on, where we could look at transcriptomes and try to understand what was going on at a tissue by tissue level, and maybe start to pick at how the genetics is being translated through these different tissues, through different functions, towards these ultimate disease-related traits. So we did a lot of eQTL analysis, and I'll skip to a key finding, and this was replicated across all the different tissues, which was when we looked at genes with very strong local eQTL, and those that have done these studies know that you can get Whopper lot scores of 150, that sort of thing, where you have local genetic variation that has a very, very strong effect on the expression of a local gene. We can explain, in many cases, a lot of the variance of that gene across the population in terms of that genetics, and that's what's shown on the y-axis everywhere here. And so just on the left here, let's focus on the local eQTL. It's shown here on the y-axis in all those tissues. And then we asked, when we try to correlate that gene's expression with all the traits, how well correlated is a given gene with any trait? We just took the maximum correlation that it could explain here. And we found overall that the genes that have a lot of variance explained by their local genetics are not the same genes that correlate well with the traits. So you can see that's a negative. I mean, I don't know that the negativity of the correlation matters here, but if you had a few transcripts that were genetically controlled locally and very well associated with the traits, you would see a bunch of stuff here, up here in the top right corners of these plots, and we don't. If we ask, what about genes that have distal genetic variation affecting their expression? That is to say, we can assign variants in that gene to somewhere else in the genome. We do get a bit of a correlation with how well those genes correlate with traits. Okay, so this is our first suggestion that it's the distal eQTL that might be more relevant to the health outcomes. And if you think about it, this probably makes sense. If you have a variant that is affecting an overall health outcome, that shouldn't just be relevant in the gene that is closest. It probably have a signal across many genes in tissues, and that's what we're seeing here. So this was step one. I sort of think of this as like expression drift. There are a lot of genes that can move a lot due to local eQTL, and the system's buffered against that. You still get a liver, you still get adipose tissue, you still get a living mouse. And so to dig into this a little more and try to understand how these are actually connected, we introduced this idea of high dimensional mediation analysis. And so this is to basically do mediation analysis, but across the entire genome, the entire transcriptome, and what we call the phenome, which is our collection of traits, at the same time. And so we basically try to force the genome to phenome, the G to P association through the entire transcriptomes. Okay. And so all that variance, like with mediation, but again, this is at the full scale of the full genome and the full transcriptome, goes through what we call TC, which is our solution of weighings of transcripts, basically, that transmit genetic information. So this still originates in genetic variation to the phenotype variation. And when we did that with our data sets across all these different tissues, we could get basically all of the variation feeding through the transcriptome. There's enough variation in the transcriptome such that we can model the genetics to phenotype correct correlations that way. And on the right, you can see if we were to permute all of the observed transcriptomes, we can't really do this very well. Our observed solution is a big outlier. It has much higher correlation between transcripts and phenotypes. And again, that's suggesting that the transcripts that maybe are moving the most aren't expressing how the genetics controls the phenotypes. And we get a slight lower than expected in terms of how the genome affects the transcripts, which again is maybe weeding out some of that very strong local variation. We then looked at that transcript matrix. These are the transcript loadings and the weights of how important each transcript is relevant to these phenome outcomes. So on the left are the collection of phenotype loadings. So that's basically saying these adipose or these metabolic traits are correlated. There they are. On the right, though, is more interesting. You're seeing how the different tissues have different loading magnitudes. And what we see is the adipose has the most genes with strongest loadings, the biggest mediator between the genetics and the outcomes. Islets are next, liver and skeletal muscle after that. We can dig into these loadings. You can think of them as, weights or, virtual expression values, whatever you want to do, but that allows us then to do an enrichment analysis. We can see which pathways are being modified by tissue. What we see is a lot of inflammation up in the adipose that's being linked through this mediation, stress response in the liver, et cetera. If you look at a given pathway, you get this absolute clarity in terms of loadings, like everything is up here in phagosome activation, right? So this you'd see in gene expression, but it comes through much, much stronger when you feed it through this must connect genetics to the phenotypes mediation analysis. Okay, now if you look at the genes with the large loadings, and that's what I'm showing here on the left, the largest by magnitude, so the most positive, most negative genes, and their loadings by tissue. So adipose wins. The top 10 in adipose beat the top 10 in any others. You can see that if we look at local heritability, it's quite low on almost all of these. That's what the blue things in this heat map mean. Whereas a lot of these have pretty good still heritability. Full-face genes are genes that have already been associated with metabolic outcomes. And so we're seeing a pretty good representation of such genes there. If we compare that to the genes just with the largest local heritability, so these are the big, the big local variants explained. Very, very weak loadings across the board, and not so many genes that have been associated with obesity. We have a threefold enrichment on the left. Okay, so again, the genes with distal heritability tend to be more informative if we want to link the genetics and the traits. So we tested this in other populations. First, we looked at a collaborative cross recombinant inbred population, where we took some CC lines, crossed them to make heterozygous F1s, but repeatable individuals. We did that, measured a number of traits, and asked how well can we predict those trait outcomes that matched our previous study, given two models. One is if we took our transcriptome loadings, and the transcriptomes through those loadings and predicted phenotype, and we saw that from adipose and liver gene expression, we could predict body weight pretty well. The other model was to take the genotypes, predict transcript levels from local genetic variation, and then predict the traits from there, and that did not work. So this is the same founder populations mixed up in a different structure, but we're losing that predictive power if we only look at local, like TWAS style prediction. And finally, let's try it in humans. If we take those loadings, that solution we got, and go to human data sets and feed human gene expression levels, and I think these were adipose, and try to predict obesity, we can sort of do it. And so this is information coming from the mouse model in these various tissues through our mediation analysis, telling us what genes are important and how to weigh them, somewhat accurately predicting what you would expect in human outcomes. These are kind of tough. There aren't a lot of samples to go within the human studies, but still somewhat promising. So to summarize, I think we're finding the transcripts that drive metabolic outcomes and really linked to traits are constrained by a lot of distal genetics. It's not just the local genetics that tells you the genes that are most important. There's a big network of interacting genes, proteins, et cetera, and those constraints can come through somehow. This sort of mediation analysis, we can see the outcome of those constraints. We don't really understand the biology of it, but it seems to be there. Genotype, local EQTL, and sort of TWAS are actually pretty poor trait predictors for independent populations, even when you're starting with the same genetic material in a mouse population. It kind of surprised us. We thought it would work better than it did. But those mediator transcripts that we inferred were actually quite predictive in the independent population, and even a little predictive in the human population. So most of this work was done by Anna Tyler in my lab, who is not here, but she is online. Matt Mahoney, who is back there, he's present. We worked with Gary Churchill and also the Keller and Addy at University of Wisconsin. Thank you. Thank you very much.
So I have a question about your transcript genomic data. Is that taken from animals after they've already developed the metabolic obesity phenotype?
Yeah, they were all 22 weeks, I think. All animals were sacrificed at the end of that study.
So for the predicted transcripts, we look to see if those are could be predictive before disease onset, like in more juvenile.
Yeah, in a sense, this population is that pre-disease prediction. So the diversity outbred mice are frankly pretty healthy. Very few of them have frank diabetes, but we are looking at variation in those relevant traits. So insulin levels in blood, glucose tolerance test results, that sort of thing. So in a way, these are all pre-diabetic, or the worst of them are pre-diabetic. And so I would say that's basically what we're modeling at the moment.
That was some of the initial data. And they showed a pretty strong sex effect. The person called it gender effect and really shot down. But did you separate that by sex and did you see any? And could we be losing signal because?
Yeah, I think we just did the sex covariate across the board in these analysis. So that's a great question. If we would have the power to stratify, we should try it and see what happens. Yes. Yeah. We have a preprint as of 2 days ago, so not published but available.
I was wondering, what about those genes that do have a strong GTPL? If you take it out, the breast out, is there still signaling the distal genome for those?
Yeah, that's a great question. We're trying that at the moment just to see what happens. But yes, our prediction is the same, that if we were to address those local effects out, it wouldn't change much. It might change a few things, which might be interesting.
So it's really the gene, the identity of the gene. It's not so much whether it's the proximal or distal component of the.
Yeah, it's the gene, and what we're finding is the distal is more important if you're thinking about how the trait variation, but it's the living of the gene. The expression of the gene is the key predictor. It's just that expression is controlled by the genetics through this mediation.
Okay. Greg, I'll catch up with you later, but just very briefly, the animals in your sample are all related to each other, right? The DL and they found complicated feature. When you do the distal, really of the animals, which would kind of control all of the variable variants. Do you do anything to get away from that? Like the fact that you can predict in other samples seems compelling, but I haven't maybe understood how you do the distal predictions. Okay, guard against castrate relatedness.
Yeah, the distal is the non-local components, right? So we're not trying to map specific distal factors. And so
all of them,
yeah,
right?
it's the entire structure.
Like yes. Okay, good. I'll talk with you later. This is really exciting.
All right, our last speaker is Katie Hall, and she is on dietary exposure of PFOs and HHS.
Morning, everyone. Thank you all for this opportunity to talk to you all today and to show you the work that we're currently doing. As mentioned, my name is Katie Hall, and I'm a research associate in Dr. Ann Quiddick's laboratory here at the Medical College of Wisconsin, where we use rat models to study the effects on metabolic syndrome. And today I will be talking to you about chronic dietary PFAS exposure on heterogeneous stock rat founder strains. First, let's talk a little bit about metabolic syndrome, or MET-S. Is it because I have my pointer on? Is that probably why? Good. Just use the arrow down here. Okay. So first let's talk about a little bit about metabolic syndrome or MET-S. MET-S results from multifunctional conditions coming together to rise your risk of serious health complications such as heart disease, hypertension, type 2 diabetes, and numerous others. To be diagnosed with MET-S, you need at least 3 of the 5 characteristic features listed here, such as central obesity, high blood pressure, and high triglycerides, low HDL cholesterol, or insulin resistance. 1/3 of all U.S. adults are considered to have MET-S. It's a complex syndrome with multiple factors with no simple answer as to what causes it. MET-S itself and all its component features are all genetic, complex genetic traits with high heritability. Unfortunately, the genetic links are mostly undetermined. Numerous genome-wide association studies have validated about 100 loci for both hypertension and obesity, but when combined, these variants only explain a small fraction of the phenotypic variation. So there's still a lot of work to do, which is where we come in. So endocrine disrupting chemicals, also known as EDCs, are natural or man-made chemicals that may mimic or interfere the body's hormones. One type of EDC is per or polyfluoroalkyl substances known as PFAS. PFAS is a forever chemical that is resistant to water, oil, heat, or stains. They have strong carboxyl and sulfonate groups that make them resistant to degradation. And there's no known natural mechanism to break them down. Because of these features, they're attractive to the food, textile, and personal care industry. They have long half-lives ranging from 2 to 5 years, depending on the type of PFAS, and there are currently over 15,000 different types, but only a few are actually studied for what the effect is on your health. PFAS can be found just about anywhere. It's in food wrappers, packaging, firefighting foam, waterproof clothing, nonstick cookware, cosmetics, basically anything that's deemed waterproof. It's even in our drinking water, and new research suggests that it is in rainwater as well. There are so many types of PFAS and so many products that in 2022, it was noted that approximately 98% of Americans have PFAS in their blood. Kind of terrifying. As you can see from the map, PFAS is a contaminant that's abundant around military sites, airports, wastewater plants, and industrial areas. We are exposed to PFAS through physical contact, consumption, and inhalation. PFAS is not metabolized in the body. It leaves slowly through time via excretion through urine, feces, menstrual fluid, and breast milk. It does cross the blood-brain barrier and has a strong binding to serum albumin and accumulates in the liver and the kidneys. Because of this, there are numerous health effects from PFAS exposure, such as eye diseases, certain cancers, thyroid disease, liver damage, and metabolic disruptions, along with developmental effects that affect the fetus, such as premature birth, low birth weight, and hormonal disturbances. There's been some research by the European Food Safety Authority that suggests a tolerable daily intake or TDI, which is how much PFAS you can consume before you start to see health effects, for 4 types of PFAS of 0.63 nanograms per kilogram body weight per day. So for a 150 pound person, that equates to about 43 nanograms of PFAS per day. So what the heck does that mean? Let's take a closer look. So here you have a typical healthy salad with vegetables, an 8-ounce piece of cod, and a nice cold glass of Wisconsin milk. This single meal alone contains 57 nanograms of PFAS, which already puts you over the recommended TDI in this one meal alone. The Environmental Research and Education Foundation estimates an exposure of 146 to 600 nanograms per day from food and dust alone. It's not to mention any additional PFAS you may be exposed to during the day, such as the yoga pants you're wearing, the makeup on your face, or the 605 million nanograms per kilogram of PFAS in the container that surrounds your Big Mac from McDonald's. All right, so genes by environment. Our lab has previously studied other endocrine disrupting chemicals and their effects on the HS founders. In 2023, Dr. Valerie Wagner headed a study where we exposed 6 of the HS founders to bisphenol F via drinking water. I don't want to go into the amazing benefits of using the HS, as we've heard that already numerous times today, and we will continue to hear about it because they're amazing. But the results show both sex and strain-specific exposure outcomes in relation to metabolic disease and endocrine disruption. Based on those findings, we designed a dietary PFAS exposure study using the HS founders. And we hypothesize that chronic PFAS exposure will have strain-specific effects towards metabolic disease and endocrine disruption. To test this hypothesis, we used 7 of the 8 HS founders. One of the founders, the WN, is extinct and is no longer available. We placed 6 of each sex per strain on either a control diet of regular chow or a PFAS diet comprised of a regular chow plus 30 mg per kilogram PFAS. The type of PFAS we decided to use is called perfluorooctane sulfonic acid potassium salt, or PFOS. We chose this PFAS because it's heavily used in industry and literature reviews suggest detrimental effects. It's important to note that the HS founders that we are using are a different substrain than the original HS founders. Once the rats are born, they are weaned at 3 weeks of age and placed in nuclear magnetic resonance, or NMR, where their body composition is measured. They are then placed in metabolic cages where we collect urine and feces for further metabolic measurements. Their diet is then started at that time. At 13 weeks of age, we measure glucose tolerance via an intraperitoneal glucose tolerance test, or IPGTT. We take lipid measurements. At 14 weeks, they are placed back into NMR and metabolic cages for the same measurements that were taken at 3 weeks of age. And then their tissues are harvested at 5 weeks of age. From week 3 until harvest, their body weights and food weights are measured weekly. And to account for any seasonal or litter-specific variants that we might have, we tried to spread out the enrollment of each of the strains and exposures over the course of a few months. This is an ongoing study that we're currently doing, and for time's sake, I will only be discussing the current results for just the males and not the females. There was no difference in food consumption between the controls or PFAS animals across all 7 strains. The PFAS food was palatable, and there was no reason to believe that there were any differences in eating behaviors between the 2 groups. Exposed rats ate between 400 to 600,000 nanograms of PFAS per day, and their average PFAS dose, which was between 2,500 and 3,000 nanograms per kilogram body weight per day. Using the animal to human dosimetric adjustment factor equation, this PFAS dose equates to about 600 nanograms per kilogram body weight per day, PFAS in a 150-pound human. This is a few orders of magnitude above the recommended TDI of 0.63 nanograms per kilogram body weight per day that I mentioned earlier. We realize this is a high dose. However, when designing this project, we based our exposure on numerous published papers on PFAS rodent exposure, and our doses is actually on the lower end of concentration. Plus, we wanted to be sure to uncover any genetic differences. Despite having consumed the same amount of diet, the exposed ACIs had a significantly lower body weight gain of about 8% during the duration of the study. NMR revealed that the exposed fishers had approximately 29% less overall body fat than the controls. Digging a bit deeper into adiposity, you can see this decrease in overall body fat in the fisher is also present in their visceral fat, which is a combination of the gonadal and perirenal white adipose tissue, along with the fisher at a decreased percent of 28, and the exposed MRs and WKYs also show a significant decrease of 17% and 34% respectively in visceral fat. The same trend in the fisher and MR continue with subcutaneous fat, or inguinal white adipose tissue, with a decreased percent of 22% in the fisher and 14% in the MR. However, this is no longer this difference is no longer exposed in the WKYs. We believe this is because the WKY is more of an apple-shaped strain where they store fat in their abdomen, which is a common trait of metabolic syndrome, instead of a pear-shaped strain like the fisher or MR, where they store fat in their hips and their thighs. Moving on to serum triglycerides, which is a measure of circulating fat in the body, it appears that the MR strain is greatly affected, showing the exposure grouping having a 67% drop in triglycerides. There have been a few studies that show long-change PFAS, which our PFAS is, affects glycerol phospholipid metabolism, which results in lower triglycerides, which is clearly being shown here in the MR. As a side note, we do not see these elevated levels in the MR female control, so this might be a sex variation within the MR itself. When looking at tissue differences, every strain except for the buffalo, because right now we have low numbers, show a significant increase in liver size of approximately 25 to 51%, depending on the strain. This could be due to the toxic amount of PFAS the rats are consuming, or potentially due to non-alcoholic fatty liver disease, fibrosis, or inflammation. Both the exposed ACIs and WKY have a decreased heart size of around 11%. This decrease in size is perhaps due to decreased wall thickness of the left ventricle, which is indicative of hypertension. I know I threw a lot at you, so just a quick review here shows that the ACI and WKY have a decreased heart size. All strains but the buffalo, because we have low numbers at this point, have increased livers. Fisher, MR, and WKY have decreased fat, and the ACI have a decreased body weight gain. So based on the significant differences in the phenotypes presented, you can conclude that there are strain-dependent effects from PFAS exposure. There's still a lot of work to be done, especially at the benchtop. So we plan on investigating the increased liver size to see if non-alcoholic fatty liver disease is present. So we wish to take a look at liver lipids and enzymes along with histology to look for fibrosis and lipid accumulation. We would like to perform RNA extraction and qPCR and any genes associated with fatty acid metabolism and liver injury, measure serum cholesterol, look at hormone effects, particularly possibly in the thyroid, measure T3 and T4, perhaps record blood pressures to measure any cardiovascular differences since we see that decrease in heart, and of course repeat the study in our amazing HS rats so we can run a genome-wide association study to identify risks that will possibly then translate to human studies. So I'd like to thank everybody here, especially everyone in the Quiddick lab, Dr. Ann Quiddick, our fearless leader, Samuel Hernandez, who's our MSTP student, and Dorian Pina, who was our Aspire and Spur student this summer and last summer. And of course, everyone listed below, Dr. John Rihu at the CRMPC, Dr. Luna Donnell from the HRDP, Lynn Malloy, who's our rat dealer, and gave me supplied all the rats that we need for this study, and of course our funding through the AHW and the HRD. Thank you very much.
Surprised that a lot of the metabolic markers were actually healthier, right? Lower triglycerides, lower fat pads, and I think make it hurt.
Right. I mean, by the sound of it, you know, the decreased weight gain and the lower fat, I mean, it sounds great, right? Let's all have PFAS. Woo-hoo. Don't do that. Yeah, it was very surprising, and we don't know if that's just because it's just that unhealthy for you, and you have such a toxicity in your liver that your whole body is just like, whoa, this is not healthy. I need to start shutting some stuff down, and potentially that's why. But yeah, it was interesting to see how actually healthy, in a sense, PFAS makes you.
In discussions of actually all of these, Obesity and Metabolism syndrome or related, does any of you study thyroid function in terms of the allergy itself, knowing that actually thyroid very much affects almost all of those phenotypes you're talking about?
Correct. That other study that I previously had on that Dr. Valerie Wagner did, for BPF exposure in drinking water, she did find significant differences in thyroid expression of T3 and T4. So yeah, that is a very common thing with metabolic syndromes to have thyroid issues.
I think there were two strains, the ACI and the
I don't remember.
That had that thyroid function differences. One was like hypothyroid and one was hyper. I'm in genetics yet. So just look at. Hopefully, if we can do a GWAS and HS, we'll look at that for sure, right? Because we know that there are strain differences, but we haven't done. Sorry.
No, you're good.
I have a question related to the, so are the independent drugs chemical, do they also produce this decrease in triglycerides and body weight, or it is specific for the PFAS from nutrition?
So the previous study also that I showed on the slides, I believe with the BPF, she did not see differences in overall body weight. And as far as anything besides BPF, I'm not really sure. Go ahead, Abe.
Yeah, great. Really nice talk. The PFAS, you say isn't clear during when it's kind of excreted, but not metabolized. But did you make any attempt to measure PFAS levels in these animals that were they been measured?
That's a really good question. So basically to do that, we don't have the equipment in the lab to do that. That would all be through mass spec. So I think potentially another thing we could add to our list is actually verify how much PFAS is in the diet to begin with. Another thing to think about is our control diet. Is it really a control diet? I mean, there's probably some small amount of PFAS in the control diet. So that would be interesting to do. And then we did collect blood plasma during IPGTTs and some other points along the line. So that would be the next move, is to take that into mass spec and see exactly how much exact PFAS these guys are getting. So, yeah.
Question. So for when you're trying to predict differences for the strains, I think it'd be really cool to do a next size so you can compare probably the magnitude versus like 4 stars versus 1 star. The significance is usually just significant. And then for in terms of this being like something that accumulates, have you guys, or do you have any plans to look at age effects?
Not really. This was just a pilot to kind of get our foot in the door, but those are all really good suggestions. Kind of our bread and butter is to bring them out to 15 weeks. That's kind of just been our endpoints, but that would be something unique to definitely venture into, is bring some out to 10, maybe 15, go to 20. And yeah, that would be very interesting.
We have 9 minutes left on our discussion, so if you have any questions for other speakers, feel free to voice them now. I have a question. Okay, did you see any difference in sexual maturation in this animal? Probably adolescent to development. This is hormone disrupted.
We didn't really look at that. We haven't, I mean, as far as, yeah, we haven't really ventured down that road yet. Like I said, we're still really early on this. We're still working through it, but that would be another thing that we can look at as well. And we actually, come to think of it, we didn't take any testes or ovaries, reproductive type organs. So that would actually be an extension. Hopefully is to get this funded again, repeat it, and then that would be something that we would want to do.
No questions on any of the other talks? Not, I think it's about lunchtime. I'm hoping it's actually already served up across the hall. So, thank you for everyone. It was a great morning. Thank you to the speakers, and we're going to get outside.